Hello everyone, today I'm going to be discussing how field realistic exposure to imidacloprid affects the foraging trip duration of the western honeybee Apis mellifera. So imidacloprid is one of the most popular pesticides and high doses are of course lethal to insects. However, honeybees are often exposed to sublethal doses when they collect nectar and pollen from treated plants. These concentrations don't kill the bees, but they can interfere with their foraging behavior. This presentation looks at whether field realistic imidacloprid exposure increases the duration of individual foraging trips for honeybees. Before discussing imidacloprid specifically, it's important to understand why foraging trip duration is an important measurement in honeybee research. So a typical foraging trip begins when a worker bee leaves the hive to search for nectar or pollen. And after locating a food source, the bee collects the resource, returns to the hive, unloads it to other workers, and then repeats the process many times throughout the day. So under normal conditions, these trips are relatively efficient and predictable. So healthy foragers are able to navigate using visual landmarks, the position of the sun, and learn cues from previous trips. Because of this, researchers can measure how long a bee is away from the hive and use that as an indicator of how efficiently it's foraging. If bees begin taking significantly longer trips, it may indicate that they're having difficulty navigating, locating resources, or returning to the hive. Even if the bees survive pesticide exposure, spending more time on each trip means they complete fewer trips throughout the day and ultimately bring less food back to the colony. This makes foraging trip duration a valuable way to measure the sublethal effects of pesticides like imidacloprid. To understand why imidacloprid affects foraging, it's important to first understand how complex honeybee foraging really is. The model organism used in all the studies I'll discuss is Apis mellifera, commonly known as the western honeybee. Honeybees are one of the most important pollinators in both natural ecosystems and agriculture, making them an excellent model for studying the effects of pesticides on insect behavior. A successful foraging trip depends on several different neurological processes working together. First, bees use olfaction, or their sense of smell, to identify flowers and recognize rewarding food sources. They also rely on learning and memory to remember where those sources are to locate and return to them efficiently. Once a bee has collected nectar or pollen, it must navigate back to the hive using visual landmarks, the position of the sun, and other environmental cues. Finally, successful foragers communicate the location of profitable food sources to nestmates through the waggle dance, allowing the colony to forage more efficiently as a whole. Because all of these behaviors depend on proper nervous system function, any chemical that interferes with neural signaling has the potential to reduce foraging efficiency. This is why researchers expected imidacloprid, which acts directly on insect nerve cells, to affect foraging trip duration. Now that we've looked at how honeybees forage, we can better understand why imidacloprid may interfere with this behavior. So imidacloprid belongs to a class of insecticides known as neonicotinoids, which are among the most widely used systemic pesticides in agriculture. Because they are systemic, the chemicals absorbed throughout the plant and can be present in pollen and nectar that honeybees collect while foraging. Imidacloprid works by binding to nicotinic acetylcholine receptors, or NACHRs, in the insect nervous system. Normally, these receptors help transmit nerve signals that control behaviors such as learning, memory, movement, and navigation. However, when imidacloprid binds to these receptors, it causes continuous stimulation of the neurons. This overstimulation disrupts normal nerve signaling and can impair the bee's ability to process information and respond appropriately to its environment. In this presentation, I use the term field realistic exposure to describe pesticide concentrations that honeybees are actually likely to encounter while foraging in agricultural landscapes, rather than unrealistically high doses used only in laboratory toxicity studies. After reviewing the biology of honeybee foraging and the mechanism of action of imidacloprid, I can now introduce the specific question that this presentation aims to answer. My research question is, how does exposure to field realistic levels of imidacloprid affect the foraging trip duration of Apis mellifera? Based on what we know about how imidacloprid disrupts normal nerve signaling, my hypothesis is that honeybees exposed to field realistic concentrations of imidacloprid will require more time to complete their foraging trips than unexposed bees. The reasoning behind this hypothesis is that successful foraging depends on learning, memory, navigation, and sensory processing. If imidacloprid interferes with any of these neurological functions, bees may take longer to locate food, become less efficient at returning to the hive, or even fail to return at all. The first study I examined was conducted by Wang and colleagues in 2025. Unlike laboratory studies that often use extremely high pesticide doses, this study exposed honeybee colonies to field-realistic concentrations of imidacloprid. The researchers wanted to determine whether these low concentrations affected the bees' ability to collect food under normal field conditions. So to do this, they monitored colonies using an automated imaging system that identified whether returning foragers were carrying pollen. This allowed them to compare the foraging activity of colonies exposed to imidacloprid with untreated control colonies over time. Looking at the figure, the most important result is that colonies exposed to imidacloprid returned with significantly fewer pollen foragers than the control colonies. In contrast, nectar foraging changed much less. This suggests that pollen collection is particularly sensitive to imidacloprid exposure. Although this study did not directly measure individual foraging trip duration, it demonstrates that field-realistic imidacloprid exposure reduces overall foraging efficiency. If fewer pollen foragers are successfully returning to the hive, the colony collects fewer nutritional resources needed for brood development and long-term colony growth. This study provides the first piece of evidence that even sublethal levels of imidacloprid can alter normal honeybee foraging behavior under realistic environmental conditions. The second study, conducted by Schneider and colleagues in 2012, directly examined how sublethal imidacloprid exposure affects the duration of individual honeybee foraging trips. So, in this experiment, the researchers attached small RFID tags to individual worker bees, and these tags automatically recorded the exact time each bee left the hive and the exact time it returned. So before the bees began foraging, they were fed known sublethal doses of imidacloprid, while a control group received no pesticide. And this allowed the researchers to compare the foraging behavior of exposed and unexposed bees under the same conditions. And looking at the figure, the y-axis represents the amount of time bees spent away from the hive during each foraging trip. Across the experimental group, bees exposed to imidacloprid generally spent more time completing their trips than the untreated control bees did. The statistical analyses shown on the figure indicate that these differences were significant for several of the treatment groups. Three of the four treatment groups showed a high significance level with these results. These results also support the hypothesis that even the low non-lethal concentrations of our imidacloprid interfere with the neurological processes required for efficient foraging. So, because honeybees rely on learning, memory, and navigation to quickly locate food and return to the hive, disruption of these systems causes bees to spend more time away from the colony. And this study provides the strongest direct evidence in my presentation because it measures the exact response variable in my research question. Well, the previous figure showed that imidacloprid increased the amount of time bees spent on individual foraging trips. This second figure from the same study helps explain why that matters at the colony level. So, using the same RFID tracking system, the researchers measured the number of foraging trips completed by each bee per day after exposure to sublethal doses of imidacloprid. Because every departure and return was automatically recorded, they could accurately compare the activity of treated bees with untreated controls. So, looking at the data, bees exposed to imidacloprid completed fewer successful foraging trips per day than the control bees in several of the experimental trials. This is exactly what we would expect if individual trips are taking longer. When each trip requires more time, bees have fewer opportunities to leave the hive, collect food, and return before the end of the day. So, this finding is important because it connects to individual behavior to colony performance. A single bee taking an extra few minutes to complete a trip may not seem significant, but when hundreds or even thousands of foragers are making fewer trips each day, the colony brings back substantially less nectar and pollen over time. So, together, the two Schneider experiments provide strong evidence that sublethal imidacloprid exposure not only increases foraging trip duration, but also reduces the overall efficiency of honeybee foraging. The evidence presented in this presentation consistently supports the hypothesis that field realistic exposure to imidacloprid impairs honeybee foraging behavior. So the first study demonstrated that colonies exposed to field realistic concentrations of imidacloprid returned with fewer pollen foragers, indicating a reduction in overall foraging efficiency. The second study used RFID tracking to show that individual bees exposed to sublethal doses of imidacloprid spent significantly longer completing their foraging trips than untreated bees. And finally, the third experiment found that these bees are also completed fewer successful foraging trips each day, further reducing the colony's ability to collect food. So together, these findings suggest that even when imidacloprid does not immediately kill honeybees, it can still have important behavioral effects by disrupting learning, navigation, and other neurological processes required for efficient foraging. One limitation of these studies, albeit, is that they primarily measured short-term behavioral responses. So additional long-term field studies are needed to determine how these behavioral changes affect colony growth, reproduction, overwinter survival, and other things over extended periods. The significance of these findings goes beyond the bees that were studied. So honeybees are arguably the most important pollinators in the world, and they contribute to the reproduction of a bunch of wild plants as well as a ton of agricultural crops. Because colonies depend on thousands of efficient foraging trips every day, even small behavioral changes can have meaningful consequences over time. One of the most important takeaways from this research is that pesticides do not have to kill bees to negatively affect colony health. The studies I presented showed that field realistic levels of imidacloprid can alter behavior by increasing foraging trip duration and reducing the number of successful foraging trips completed each day. As a result, colonies may collect less nectar and pollen, leaving fewer nutritional resources available for brood development and colony maintenance. These findings also have important implications for pesticide risk assessment. Traditionally, pesticide safety is focused heavily on bee mortality. However, this research demonstrates that sublethal behavioral effects, such as impaired navigation and reduced foraging efficiency, should also be considered when evaluating the impact of pesticides on pollinator populations.